All right, thanks for coming out. My name is Trevor Pitcher from the University of Windsor at the Great Lakes Institute for Environmental Research. And I want to start off the talk, not about the talk, but to invite any of you that have any needs for research facilities to come visit Gleer sometime in Windsor, Ontario, which is right across the border from Detroit for you Americans. And we are, as you might guess, north of Detroit, which is kind of unusual. You'll understand that joke is from Detroit. Now, I'm actually here today. I paid a little bit extra for the program to cut me right between lunch and the next coffee break to make sure you're at your sleepiest, so I'll try to speak up a little bit more. I actually read my title on the way here in a plane, and I actually realized that the reason my wife wouldn't even watch my talk is because induction's not that exciting. So what I did is I stripped down some of the data slides from the induction work, and I'm going to actually broaden out the talk and keep it on time, and talk a little bit more about some of the work we're doing as part of a large team, and I want to emphasize part of a large team working on restoration efforts to restore bloaters or Coregonid Symposium to Lake Ontario. My lab, specifically the Pitcher lab, we work mostly on reproductive ecology questions for reintroductions, whether that's Atlantic salmon or sturgeon or red-side dace. So we're privileged to become part of this team and essentially come in and start working on some of the issues related to reducing broodstock and some of the reproductive issues that were challenging them at the start. I do want to acknowledge the student, Alex Priscello, who did his master's on this topic, and then collaborators who, like I said, are more numerous than this list, but include Jennifer Smith, Ryan Zhang, who's here today, Tim Drew, who's here also, Kevin, I think, is here, or he might have skipped out for this talk. And, oh, he's here, unfortunately, and myself. And as I mentioned, we're just one cog in a big set of wheels doing this work, and I want to acknowledge, obviously, a bunch of our collaborators who are here in the audience as well. So what we challenge, the challenges faced in developing a broodstock production, there are numerous, but the three I'm going to talk about today really lie in these three areas here. One is the reduced frequency of gamete expression from these bloaters. I'm going to just refer to them as bloaters. Due to the fact that in the captive setting, their environmental cues are not the same as in the wild. We hypothesize that may be holding back their reproductive capabilities. Number two, males and females often produce free-flowing gametes occasionally, but when they do, they're often asynchronous, so we have a timing issue. So the first one is they don't really produce enough eggs in the captivity. Second one is when they do, the males and females are off time. And the third one is that we also want to understand, as the broodstock becomes less wild in terms of being domesticated over time in the hatchery setting, what are the potential effects of domestication on egg quality parameters? And by understanding that, can we mitigate some of these issues when we release these fish back out into the wild? So fortunately for me, there's lots of problems with reproductive ecology, so I've got lots of jobs. So the three projects I'll talk about today very briefly each is the first one is the idea that we used luteinizing hormone, or LHRHa, essentially an analog to essentially induce the fish, and I'll show you some of the data about that. And that's to reconcile some of the issues where there is no environmental cues to help the fish decide when to spawn. We're going to convince them to spawn with hormones. Second is cryopreservation of bloater sperm. As I mentioned earlier, they don't often synchronize male and female reproduction, and as a consequence, we can actually cryopreserve the male sperm, and when females come online, we have a ready supply of milk so that we don't miss that peak spawning season, and as such have less output. Number three, as I mentioned, is we had a recent fortunate event where they were collecting some wild bloaters from Lake Michigan, and we got them to send us some wild fish, and we were able to compare the egg quality between the hatchery broodstock that we've been working with and the wild fish. And by understanding this domestication, we may be able to, like as I mentioned, mitigate some of the problems that have been common with salmonid captive breeding on the west coast of North America. So let me start with project number one. This is a standard issue, essentially. There's a bunch of different drugs you can use to essentially convince fish to spawn when you want them to. And this is referred to as induction for the purposes of gamete expression. And we use a variety of reproductive hormone analogs in my lab. We use a variety of kinds. In this particular case, we use LHRHa. And the idea, if you're not familiar with this, is essentially to kickstart the hormonal cascade. And I'll show you that in a second. Now, the reason we chose LHRHa in this case is because it's commonly used for many species, including a variety of other coregonids. And importantly, it has a low species specificity, meaning it works almost on all fish we work on, and it's relatively speaking quite effective in other species. So we decided to try this first to see if it would work for the bloaters. If you're not familiar with the hormone cascade, it's fairly straightforward. Generally speaking, and with gross generalizations here for time, essentially there's a suite of environmental cues that the fish perceive, whether it's temperature related, light related, we saw some talks about light earlier, temperature or water quality issues, That essentially kickstarts the hypothalamus in the brain system, which initiates a hormone cascade through the pituitary gonads and ultimately leading to the production of either sperm or eggs, depending on your sex. And the real challenge in the hatchery system often is that these environmental cues, these arrows that were there before are replaced by X's now, those cues are absent or altered in the captive setting, and as such, the reproductive capability of these fish in the captive broodstock is challenging. So what we did in this first project of 3, and this is part of a larger group of efforts we're doing to reproduce these fish, is that we essentially injected LHRHa into the system through peritoneal cavity, which I can explain later in detail. And essentially what it does is it kickstarts the whole process. Some drugs, if you're familiar with them, will actually cascade from the brain down. This one actually kicks in, essentially skips the bypass the brain system and essentially gets down to the pituitary right away. So it's a very powerful drug. And what we did then is do an experiment that essentially wanted to ask the question, did the LHRH essentially help the females produce free-flowing gametes? And so at the start of this process, not many females were essentially producing viable eggs. So we started measuring whether or not females could express the eggs after the injection or not. And importantly, if females did produce the eggs, we started getting interested in the quality of those eggs based on the injections of LHRHa. So the experimental design is fairly classic. Alex Marcello and his team at M&R did this work where they essentially had 2 controls. Very importantly for you if you're doing some of these injections, they had a control which is a straight up where there's no treatment whatsoever, you just handle them but nothing else. And this is really important. They also did a sham control where they actually injected the medium, which I know you can't read, but it says saline here. And so they did the injection of the saline with nothing else and the handling stress. So we really wanted to get a better feel for what the handling stress and the injections were like. And then we had 2 dose levels. Their numbers aren't really important for today's talk, but just know that there's a low dose level and a high dose level. And when you inject LHRHa, you often have an initial injection followed up by what's called a resolving injection. So all fish got these injections if they were part of the sham, the low dose or high dose. And essentially we were able to follow up by returning these fish to the brood tank. They were all pit tagged, so we could follow them through the entire process. And of course, eventually the students every week or so were assessing whether or not there was a presence or the absence of free-flowing sperm or eggs, depending on the sex of the species of the individual. And we did this in 2 time periods. We did this in December and in January, and our sample size for December is about 692, and in January 640. So we had real power to detect the differences amongst these treatments if it existed. The straightforward hypotheses you can imagine already is, of course, we are hoping, or hypothesizing, sorry, that we would promote gamete production, so the high dose would be better than the low, which would be the same as the sham and the control. We did a fairly straightforward analysis for this talk today, although the publication will be a little more sophisticated, where we measured presence absence of the spawning using the chi-square test to see if it was significant. And then, of course, we also looked at some egg quality parameters. I'll only show you a subset of the data to keep you awake for the afternoon. And essentially the idea is that we can look at whether or not the quality of the gametes produced through injection was as good as those that were excised from the individuals that weren't injected. So here's your first data figure. Essentially what you can see on the y-axis is the proportion of individuals that produce gametes, and on the x-axis is the treatment group that I mentioned earlier, the control, the sham control, the low and high. The black bars on your left are the males, the dashed bars on the right are the females. And the real take-home message is that the LHRH, in a nutshell, had a profound effect on females. It really almost doubled the efficacy of gamete expression. But for males, it actually ended up showing a negative feedback loop. We have other data showing that not only does it not really work well, but another time period that I mentioned in January, it actually suppressed male sperm expression. We also looked at egg quality parameters. This is just one. This is the diameter in the same treatments I mentioned earlier. And then on the far right, we had a wild sampling from that same year. And the take-home message, once again, in a nutshell, is that the egg quality of those that produced free-flowing eggs after the LHRH was significantly better than those where we had to excise them, where the female wasn't expressing the eggs. And then if you look at the wild sample, the eggs in this particular year class were about the same as the wild, although the body size between these 2 groups, which I'll say for later, is significantly different. So I'll just move on to project 2 in a timely manner. So we also developed this cryopreservation method, which is technically quite complicated, to essentially work on these bloaters. And the idea is that we essentially poach some of the data from other species, such as their cousins. And what the idea is that we wanted to look for guidance for some of these variables that affect cryopreservation success. If you've never done cryopreservation, this is the classic trial and error. This is where the student just tries and tries and tries and tries and tries and tries. And when they feel like throwing up, you tell them try again. So the idea is that Alex and his team spent hours and hours doing every permutation possible, where they essentially had these cryo straws. You then pull up the milt. You then essentially seal it in a different cryo. So each of these little marks represents a different treatment. You throw it in minus 196 degrees Celsius, which is kind of cold. And then the idea is that you look at which of the chemicals protects the sperm from the thaw and freeze damage. And the gist of it, I'll save you this expense, is that we figured out which of the effects were toxic or not, and our next step is actually to save the sperm and then come back 2 or 3 or 4 or 15 years later and reanimate that sperm and look at the effects. The last project, which I'll wrap up very quickly, was comparing the wild versus hatchery egg quality. So we had a sample of eggs, thanks to Roger and the other teams on the boat, send us fish from the wild and then fish from the hatchery. You can see phenotypically, these are the average fish from those samples. They're quite different looking and quite different in size. And what we were interested in was whether or not the selection pressures in a fish culture setting were affecting egg quality or not. And the student we have working on this right now is measuring a variety of traits, including size, lipids, carotenoids, fatty acids, etc. We heard about earlier today, and GSI, amongst other things. And the one data slide I'll show you before I wrap up is we looked at GSI and we looked at how many eggs were produced per body mass. And if you look on your left, these are the wild fish. There's about 75 of them. And on the right, there's hatchery fish, 2 age classes, 2012 and 2013. And the take-home message is quite dramatic. On the x-axis, you can see radical differences in body size. So that's the first take-home message. On the y-axis, you can see that the correlations between the two traits is much tighter in the captive setting, which is not surprising because in the wild, obviously, as those females are coming up, their eggs are expressing as the pressure goes onto them. So the idea here is that if you look at the average GSI, it's 19% in the wild and 15 or 16% in captivity. But imagine that females have probably lost a quarter to half of their eggs on the way up. And so that's something we can't control, and yet they still have higher GSIs than the captive ones. So there's some kind of selection pressure that we have to be cognizant of potentially in this culture system, and we're learning more from this as we go through this data. So in conclusion, we sort of have 3 bullet points. 1, LHRH works well for females, but not males. And we're going to essentially follow up by doing more work on this, by essentially developing whether or not repeat spawners have this year. We're doing that this year, actually. Do repeat spawners actually show a different effect than individuals that have been only injected once? Next, cryopreservation. We're going to essentially follow through with the damage due to thawing and freezing. And more importantly, for those who are geneticists, we're also looking at epigenetic effects of freezing the sperm and looking at the offspring to see whether that has an effect. And finally, of course, we're finishing up that study on wild versus hatchery egg quality. And essentially, we looked at GSI differences very quickly, but we have a variety of other traits that may be important for hatching success and survival in the wild for these fish we're restoring to Lake Ontario. So with that, I really want to acknowledge many people that are here today, but also the students. This is Alex in the middle, there's Ryan, looking at Debonair, and many of the other students that work for fish culture, and of course, importantly, all of our partners. Not everyone's listed here, but of course, the funding sources, without which we couldn't do any of this work. So with that, I'd be happy to take a couple of questions.
So, how come your hatchery bloater is like monster bloater?
Yeah, well, the relative size, I mean, the obvious answer is that we just have a better menu to choose from, so they're obviously fed better. But the more interesting question is there eggs radically different in the hatchery setting than in the wild in terms of some of those DHAs and other things that may affect the fitness of the offspring that are produced in subsequent generations of stocking? So this is analogous to some of that work Dan Heath did, for example, in the West Coast, where captive fish let go over time sometimes had negative effects on the gamete quality. And so we're learning from our past by not making mistakes again. And fish culture and other groups are more progressive about studying these things. Now, any other questions, of course, I'll follow up later with you at coffee. All right, thanks.